This is a production of Cornell University.
I'm going to talk to you about something about, this idea of kind of going small, the sustainability of local breeding. And this is a question that really I started thinking about after coming to Hawaii, where everything is small. It's a very small regional market. And so through three vignettes here, I'm going to hopefully convince you of some of my ideas surrounding breeding for these local markets. So really, there are two hypotheses that I'm really hoping to explore. The first is that breeding for very large geographies is actually limited yield potential. That breeding for broad adaptation, and that really breeding for small geographies provides an avenue to increase revenue and build community interaction and kind of repair some historic bad practices. But let's start off with the first one. And I'm going to go back to some papers that I really enjoyed reading when I was in grad school. The first is Levin's, when he talks about when there's a difference between the environments is large compared to the tolerance for environmental diversity, the most fit population will be a mixture of specialists, right? So this is in some of his work when he's exploring fitness and fitness sets. And so really this means the most fit population in any given environment is going to be multiple, narrowly adapted varieties. So this can be displayed in a number of different ways, but really we're thinking about fitness sets. We're thinking about looking at the interactions that basically crossover interactions in our fitness between our different environments. We're looking at obviously in our environment, in our fitness is going to come along this gradient. And what Levin's proposed, right, is that if you look at many different genotypes, if we see our fitness in environment A and fitness in environment B, if we have a convex fitness set, we're going to have broad adaptation, and basically there's not going to be that much difference between the environments. But if we have a concave fitness set, local adaptation, we're going to have high environmental variance, and this is going to be what this is going to determine what's going to be fit in most populations. So at the same time that Levin's was publishing this, you have in the plant breeding literature, Finlay and Wilkinson talking about stability, right? So you have from their 1963 paper, the ability of some crop varieties to perform well over a range of environmental conditions has long been appreciated by the agronomist and plant breeder. So basically, this is your argument for we want a single broadly adapted variety, right? Right in line with the Green Revolution, and so their conceptualization of this was to plot so this is to plot many different varieties, and then to overlay in space where the different geographies lay. And in general, and hopefully all of you students who are here can appreciate that it's much easier to make figures now than it was in the 60s. But in general, this can be displayed like you'd see in your introductory plant breeding class with these two different kind of conceptualizations of fitness, right? With the idea of we want something where there's no G by E, right? Or where there's something with a predictable G by E. And so this is really where we started, and leading to the question of, what is the real benefit of local adaptation to commercially available crops? Because we have two different conceptions of stability. We have, and we want to know which one is actually winning, because plant breeding's been fantastic and ag's been fantastic over the last 100 years of increasing crop production. But are we leaving some of this yield on the table? So the first study system is corn. We took the University of Illinois corn yield trials from 1999 to 2018. And for a slight digression, for all of the students there, the idea for this came about because I just given a talk in 2018, and I got asked a question by a friend of mine, and then after we were talking about it after the seminar, and the question was based on an offhand comment I had made, saying that, well, you can think of breeding as accelerated local adaptation, and the question was from my friend was, well, can you? And so then we decided to test it, and so this University of Illinois data set was there because it was easily acceptable. And it was 230 varieties over 216 site years, and it's their yield trials. And so there are some problems, right? It's essentially can be viewed as a reciprocal transplant experiment. There's 12 locations, and obviously only the checks are at some locations. So just to make sure to do some, truth checking, the first thing we did was basically look at our increases in yield over time. Are we seeing increases? What are the yield, and what are the yield increases that we're seeing over time? And much like you'd expect, yields are increasing, on average by 0.2 megagrams per hectare per year. So now we wanted to conceptualize, how do we figure out what this local adaptation is? So we tried to partition the variants. So first, we have our effective genotype. We have our yield, and we have our site year and our site year interaction. But we want to get at, again, what is this kind of advantage you have in your most optimal environment? So we added this is home. And home is the location that is the best relative performance, right? And it's centered and scaled within the site year. And then we have, right, our unexplained measurement error, which is going to be our G by E. So in this case, right, our coefficient for is home is our home field advantage. And it allows us to say, it allows us to look at, if we're looking at this specific site, how big is the advantage of being the best yielder there? So the easiest thing is to just look at the partitioning of our variants here, and the home field advantage is about 20%. So basically about 20% of the residual G by E variation is per location predicts, is from the corn yield. So what this actually means is that you're getting a yield increase of about one megagram per hectare in your home field site. And that's a 7.6 increase relative to the mean, right? And so you can see that the home field advantage varies per year, but that we're still seeing that the home field advantage actually, while it varies, it doesn't really change per year. And then just to basically check our the truthiness, right? We did a permutation test, right? To permute our site years, and basically we're preserving the site and year effects and randomizing the genotypes, and we're not really, and we're not we're seeing basically that we're seeing our expected home field advantage. What we are seeing as well, though, is that specialization came with a trade-off. So what you're seeing here is our home field advantage and our variance in yield, right? So as we're increasing our home field advantage, we're also increasing the variability, which of course is non-desirable. But what we really saw is that this idea of we're leaving yield on the table in our local environment. And so basically it gets back to this idea of what is the conception of stability that we want. Do we want this broadly adapted where yield is going to be less, but it's going to be the same, or whether we're going to be seeing, or do we want to maximize it for a given location? But there's some obvious problems with this analysis, right? Is our definition of home field believable, or is it tautological? So are we assuming our conclusion in the premise, right? So we actually think that it's believable, because home sites, almost all of the sites are represented as home sites for some of the for some of the genotypes. And if you do a leave one out analysis, the home sites generally jump to the closest site that we had. And part of this actually led to a large debate amongst us on a lot of fitness measurements in general tend to be a little bit tautological. So that's why in the end we were okay with it. The second problem, which is another big one, is we didn't control for genetic relationships, right? We didn't have any information on the lines that were put in these trials. And we didn't have a way of getting it, because these were all commercial company cultivars that were in those yield trials. So we were excited, but we were also, you know, rightfully skeptical of some of these results. The idea that you know, this 7.6 or 1 megagram, you know, per hectare increase, if you're focusing that cultivar, we were intrigued, but we wanted to explore another system, right? So we moved to Common Bean. So we moved to Common Bean. Sorry, there's a chat here. For any line in the trials for greater than 1 year, can you determine the home site in 1 year and test in the other years? Yeah, we actually can, Jean, we should do that. We haven't done that yet. And that's a really good idea. We should do that with this Bean data set. Oh, thank you. But so this was Alice McQueen had published this great paper in genetics looking at the cooperative dry bean nursery trials. So this is actually 2,170 site years from 1981 to 2013, planted at 71 locations across Canada and the US. You can see all the sites. You can see all the sites here. And we wanted to see, okay, here's a system where we know the genetic relationships, everything's been genotyped. It's a longer time series over a wider geographic range with more sites. So we decided to try the same analysis, and there are obvious differences, but this let us get at some of the questions that we had regarding the obvious flaws in our previous analysis. So the first thing we did was, again, just make some basic checks. Let's plot our yield increases over time, which ends up being about 0.5% per year. What you're seeing in our different lines in this case are the major races of Common Bean. Durango and Mesoamerican are from a domestication event in Mexico, and Nueva Granada is a South American domestication. So in our conceptualization of this, we basically use our same quantile regression that we used before, only we're adding a couple of different terms. We actually explicitly now are trying to model the home field advantage by the different domestication events. We're also explicitly adding in covariates for the genetic structure and the relationships that we have between all of the genotypes. What we actually see, I apologize that these aren't donuts. As everyone should know, I would much prefer donuts, but less easy to display with the three different germplasm pools here. But basically what we saw is actually a much smaller home field advantage. So what we saw is basically the home site advantage accounted for a little more than 1% of that total variation, and about 6.5% of the residual variation. Still highly significant. The genetic relationships, when you include them as covariates in the model, were not actually significant and didn't explain much variation at all. So we were happy about that. But we thought it was interesting. Well, we're explaining a lower magnitude, but actually this lower magnitude was of greater importance because instead of being about a 3 to 4x difference in yield, you could in that yield gain year over year. Here, that year over year gain, the home field advantage was actually 13 times greater than that. So we were excited about that. We actually saw the same trends that we saw everywhere else, that the change in home field advantage was pretty stable over time. None of these regressions are actually significant. We did see that a different trend in terms of the home field advantage and the increasing variability here, in the sense that only for that Durango group did we see that kind of expected increase in variation, over as home field advantage increase. So we were actually pretty happy. We were able to figure out some problems with our previous analysis. We were able to look over a larger time frame, over a larger number of sites, over a larger geographic extent. And we have a very similar story where that in breeding for broad adaptation, we are potentially leaving some yield on the table, and economically significant yield on the table. But it doesn't get to the other half of that question. So what is then the size? What is local, right? A common question that I'm sure everyone here has come across. So one thing that we decided to look at was the spatial variability and heritability. So what you're looking at here is an ordinary Kriging across all of the sites except the Puerto Rico site. The Puerto Rico site is a bit of an outlier and causes weird things. So we dropped it from this particular, well, actually we dropped it from all the analysis because it behaves a bit strangely. So what you're seeing is this is actually the ordinary Kriging averaged over all 30 years. And what you're seeing in this case is actually our highest heritabilities in the regions where breeding has been most intense. Obviously that leads to some ideas as to where breeding might be most effective. But one of the interesting things about this analysis is when you look at the semivariogram at the decay in heritability, the decay in heritability is 1,000 kilometers. So, right, so about 300 miles. So looking at the decay of where selection is going to be most effective. And by most effective, I'm looking at it in terms of if I have a high heritability, I'll be able to do quite a bit more. It really, we started to get a picture of, well, our home field advantage and our ability to select is really going to be in this narrower geography, which is still a pretty broad geography of 300 miles. But we were actually quite intrigued by that, and we also started looking at, again, this idea of the variance in heritability measures relative to the magnitude of heritability, and it turns out, not unexpectedly, that about a third of the sites, about 40% of the sites are just garbage, because the variance in heritability is actually either of the same magnitude or greater than the heritability estimates themselves over time. So you're really, you can use it as a yield trial, but it's not a terribly useful selection environment because your heritability is effectively zero. But we also wanted to check kind of the truthiness and look at our heritability estimates over time. And so we did it, right? And so what you're seeing here is a sliding window of heritabilities on a line mean basis for 5 years, going from 1983 to 2011. And we wanted to see if this matched our general expectations on breeding for the germplasm. So what you're seeing here in the Mesoamerican is actually, you're seeing a decrease in heritability over time. And this matches what we expect in common bean, because what we have is there's been a decrease in genetic diversity in the Mesoamerican lines over time. They've been, and so this matches what we expect. Basically, while the mean in the population is improved, the genetic variance is decreased, so it's harder to make gains now than it was in the past. And actually, in our Andean lines here, we've seen the opposite trend. There's been a lot of introgression, from what Alice tells me, from these other germplasm pools into the Andean germplasm pool. So what we're actually seeing here is an increase in heritability because of this increased diversity. So we were happy about this, that basically it matched what our expectations are. Another analysis that I haven't done yet, but I'm really excited to do, is actually partition all of the sites over time into these 300-mile bins and see basically what type of variance we're seeing as we bin it by what that semi-variogram is going to say on what the optimal size for selection would be. So what does this actually mean? We have actually the same conclusions when explicitly controlling for genetics and domestication history. And it's a deficit of local adaptation. That's not unexpected. We expected to see broad adaptation. That's what we've selected for. But what I would tend to argue is that for some local markets, you can actually gain from explicitly shrinking that breeding program and looking at specific localities. And again, in our Andean, our yields tend to be lower, but they yield more consistently. The Mesoamericans tend to have higher yields, but are more variable. But I promised you 2 topics, didn't I? Hopefully, I've convinced you that breeding, broad adaptation, we've really seen, we've limited yield potential, and maybe we want to rethink that particular strategy, especially if you have resource constraints in the public sector. But I want to convince you that because of those resource constraints, that breeding for small geographies and markets can increase revenue, right? So let's talk about Hawaii. Let's talk about canoe plants in Hawaii. So the Polynesians arrived in Hawaii about 1,100 years ago. though. The archaeological estimates, I think, are some go back to 1,300 years ago, but I think around 1,100 is about the consensus. They were great plant breeders and great agriculturalists. They brought lots of plants, medicinal plants, kava, food plants, yam, coconut, taro, breadfruit, and sugarcane, also sweet potato. And in these large voyaging canoes that took them all over Oceania. These particular crops have a deep place in the epistemology of Hawaii and many of the Polynesian cultures. And they've also had great monetary benefits to Hawaii and other Polynesian nations. But because of the value that they've had, this has led to some problems, right? Universities have behaved badly in the past. There's been a history of biopiracy. There's been disrespect of traditional knowledge. And this work is documented by a colleague here at UH, Aurora Kagawa Viviani. If you guys are interested in her paper, I'm happy to send it to you. But basically a disrespect of traditional knowledge, and also the monetizing of traditional knowledge, both from medicinal and drug plants to the use of crop plants. And this has led to, in Hawaii, quite a bit of bad blood. The most recent case at the University of Hawaii was with the patenting of taro varieties, both some GM varieties that were ultimately destroyed, but also some conventionally bred taro varieties, which taro, again, in the epistemology of Hawaii, is incredibly important. So we wanted to kind of do something a little bit different, but this history basically, it can be capitalized, or is not capitalized, basically this idea of monetizing the traditional knowledge, because in Hawaii, things that are from Hawaii get a huge price premium, not only among locals, but also among tourists. So the approach that we basically took was a colleague of mine, Ted Radovich, has been working in Hawaii for years, and he's very involved in what's called the Waimanalo Pono Research Hui. So Waimanalo is one of our research stations, and the Pono Research Hui it's a community organization of mostly Native Hawaiians, but also many people who just grew up and lived in Hawaii, and they partner with many other organizations, and they do community outreach, they do scholarly work, and they do a lot of primary and secondary education. So what you're seeing here in this picture is a picture of the research station, and this is actually where the research Hui holds a lot of its event at the events on the station. So basically, working with them, we decided to do some breeding in traditional Hawaiian sweet potato. So that's kind of the name varieties that were associated with sweet potato production prior, basically during the time of the Hawaiian Kingdom, the Kingdom of Hawaii, and before. So again, so a student, here are some of the initial botanical sketches from the 40s of two varieties right here, Mohihi and Eleele. So some recent work in economic botany by Elizabeth Winicki and Aurora Kagawa Viviani, again, looked at basically the ethnobotany of Hawaiian sweet potatoes. So what you're seeing here on the left is just a K-means clustering of about 100 different varieties. And really what you're seeing is, if you look at the K equals 3 cluster here, you're seeing that a lot of the Hawaiian individuals here, sorry, I'm trying to draw a circle, but it's not letting me there, that the Hawaiian germplasm is unique, right? It's basically, it's distinct from what's in the USDA collection. It's distinct from a lot of other collections. So there is considerable value to the to having the Hawaiian names, but also to breeding only within this particular group and not doing intergressions. In terms of the idea of Hawaiian heritage varieties. So basically very standard breeding. Hawaiian varieties were put in a polycross block. Yeah, all the F1s were grown out. We did initial selections. This initial selection was interesting. So this was done. All the plants were dug up. They were all laid out, all from F1 hills. And the community helped judge what was, so it's basically community selection. We like this shape. We don't like this color. This one, we don't like the leaves. Lots of different traits. Some that I wouldn't have thought were important, but were, right? So this led to basically of the several hundred F1s, we ended up with 16 that the community really liked. We grew them out in a specific trial and a series of trials. Actually, these were the ones. And against basically our maternal parent, which was Mohihi, and the commercial check here, which is Okinawa. We had we had 3 that performed as well or better than the check. And then everyone just loves this HM17. So HM stands for Hapa Mohihi, because we only kept track of our maternal parent Mohihi, Hapa being mixed. So for whatever reason, the people really love this HM17. So even though it is a terrible yielder, we're keeping it around because the community really liked it. And basically, we can get a huge price premium for Hawaiian heritage varieties in the fresh market, and what ends up happening is that we can build a tremendous amount of goodwill with the community because they're involved with a specific species that they like. So that's important to the community. So our winners were this HM34, which looks very, very similar to Okinawa, only with a difference in skin color. HM26 is interesting because it's a monster, but they just tremendous yield, doesn't taste very good, but it's used for brewing. It's used to make a sweet potato beer. So that's where people are interested in that one. Our lines 32 and 17. I happen to just really like 32, that really nice purple color. That kind of unique peach for that HM17 is, I think, why people have really liked it. But I'm not a big fan of that one personally. The more interesting piece is how we decided to do a release policy. So there's actually scheduled for the beginning of May, or the beginning of May. There's going to be a community tasting and naming gathering with the Research Hui. So there's going to be sweet potato cooked in many different ways, along with the sweet potato beer. And the royalty plan is something quite different than what you'd expect for a normal variety release. So the KKNOW is the nonprofit that the Waimanalo Research Hui is under. And basically the royalty plan is to have the royalties go to the community organization that has been helping us with the selection. 50% is going to cover ongoing community activities, which include maintaining the lines at the station, and then 25% would go to directly to the station where all the breeding activities were taking place. So the idea here is really a new royalty, to have a royalty plan that acknowledges the basically participatory nature of this. It's unclear how much revenue will be generated, but it certainly, regardless of how much revenue, it has been great as a community building activity, and also as a recruitment activity, because there are, a lot of kids who actually participate in the on-station activities. So what can we take away from these three vignettes? Bean, corn, sweet potato, 3 different crops, but hopefully, kind of one story, right? The story is that through design, right, through selecting for broad adaptation, we've left yield on the table. We can reclaim that yield through having more targeted breeding programs. There are certainly large markets for niche, for regional and specific crops. In this case, I would say the Hawaiian sweet potato. But I think we have a lot of questions, right? How are we going to deploy populations of crops? How are we going to perform in the case of the sweet potato, which would end up being a lot of small holdings, on very heterogeneous environments? How are they going to perform in kind of those low capital systems? How can you leverage, if we're saying that we're having very strong local yield effects, right? How are we going to leverage site-specific management in higher capital systems? And with the increased variance that's obviously seen with the increased variance that we see with the increased local adaptation, how is that going to change the capital investment, and how is that going to perform, reliably in more unstable environments, right, and climates? So hopefully I've convinced you, right, that that local has a lot of benefits, especially for very small markets like Hawaii. So with that, right, I'd like to thank all the people who work on this. Brian Runk, Tom Kono, Patrick Ewing, Alice, Colin Curry, Ted Radovich, Todd Anderson, JP Bingham, the Waimanalo Research Hui, lots of different organizations, Texas, CIAT, University of Minnesota, Oregon State, Jeff and Lex for helping us think through some of these things. And so hopefully you guys believe that you can get that home field advantage and really improve yield in some of these specific circumstances. So with that, hopefully you guys have some questions, and I'm looking forward to talking.
So I was curious. One of the ways that's really common to assess local adaptation is to sort of correlate performances with environmental variables like climate or something like that. So yeah, that can be a lot more nuanced than home field advantage. So did you look at that?
Yeah. So for the corn we did, right, and so you can you saw a rainfall gradient. Right, so it's a precipitation gradient, and you saw different cultivars perform better in the different precipitation regimes. when you so basically, yeah, so we haven't done it yet for the bean, but I can tell you what we basically see there, I haven't formally done it there, but what you see is a temperature gradient. It's a latitudinal gradient, right? That probably also has to do with flowering time, but it's very much a temperature and a seasonality thing there. So I think you're right. And so what I would say is the question is one of responsiveness. How quickly can you respond to a potential change? And so, yeah, so that's what I would say. Hopefully that answers it, Martin.
Yeah, and I think that actually leads right into a question Ed put in the chat. Ed, did you want to?
Oh, yeah, I was, Mickey really enjoyed it, but I was, in the face of climate change, you know, as, you know, we may be moving to a period where things are moving 50 miles to 100 miles of adaptation per decade. How do you think about balancing out those types of things?
Yeah, so I think a lot of it depends on what scale you decide you want to work at and what is your specific question. So I know that that's an unhelpful answer and I apologize, but I think the reason breeding for broad adaptation is such a successful strategy is because it is that conception of stability where you're trying to eliminate that genotype by environment interaction hedges all your bets, right? And I think the conception where we're leaving yield on the table and you have a smaller regional breeding program is, I think if you're moving in a decade for some of these annual crops, I don't think that's a problem, right? Where you can, I mean, I guess I'm thinking of chili pepper, right? I can get 4 generations in a year, right? So if I want to move that, it's possible. Obviously much harder for longer term perennials, you know, where they're in the ground for 10 years or something. But I think in some ways you could, again, poke holes in this particular analysis by saying it's kind of it's a problem that is based on kind of having an embarrassment of riches, right? Where because you're worried about completely optimizing yield, you're not worried so much about hedging. Yeah, so I think, you know, like a lot of things, you're going to see a mix of strategies depending on what the most pressing concern is for your geography. I would also argue that, you know, again, a decay of 300 miles is still a pretty big area. So that you still have quite a bit of wiggle room. So I have you partitioned stability across locations and stability across years? Does home field advantage influence both? Yes, so we definitely have partitioned stability across years. I don't remember doing it specifically for locations. No, home field advantage didn't really impact that, which surprised me. The home field advantage that was basically the same in bad years and good years. That the average home field advantage across sites, we should really do that by sites, because I would bet by sites that's not the case. I would bet specific sites when they have a bad year. So for the average site, it doesn't, but for the individual site, it most certainly would. When you have a bad year, the home field advantage is not going to be there. So hopefully, Kelly, that answered your question. So Jean-Luc, how do you account for the geographic gradient and heritability? Sorry, the chat just jumped on me. The geographic gradient heritability for common breed. Breeding programs get better as you move from the southwest to the northwest, so lower error variance. Northwest further away from the environments of origin of being, and so novel variation revealed in new environments. So the way that I actually think about this is that a lot of the sites, a lot of the original breeding was done in the Northwest. And this big network of sites, I think what we ended up seeing is that the breeding programs in the Northwest were better funded and just did do better. So I think that that is accurate. Yeah, and so this has been with the Northwest being further away from the environments of origin. We've been so if anyone has good ideas on that side, please let me know. I think what Alice and I and Patrick have been talking about is that might be that's also where a lot of the Mesoamerican bean breeding is being done, and so we're wondering if that might be one reason why we're seeing a larger bottleneck there, basically in domestication, because you've selected for individuals that work there, and so I think that that might have something to do with it, but it's not entirely clear, and it's one of the questions we're trying to answer. But yeah, I think that's really cool. So so following on from Arcadio, I apologize if I'm not pronouncing that correctly. Following on Jean-Luc's question, could the geographic gradient on heritability be a reflection of environmental variability? So the northern locations experiencing less disturbing environment fluctuations due to the shorter potential growing season. That's a good question. I think that that's probably not the case. I think the reason we're seeing a gradient in the heritability and that decreasing gradient is that there is actually significant differences on a year for as you do that sliding window. There are significant differences in year. I think that it has more to do with I think it has more to do with trial construction and the error associated with the individual trials, just looking at the way looking at the variances by site and by over time. So I think I don't think it has to do so much with the environmental fluctuations. That would be really hard to test, but it'd be really cool. So the way that I would conceptualize doing that is you'd actually have to get seasonality data and look at planting and harvest dates, and then you would actually take the environmental data associated with the growing season and then partition that out as an additional covariate and see what that relationship would be. So that might be possible. I'll ask Alice if we have those dates, because that'd be really cool. So the more elite breeding the germplasm has been through, the more you expect low differentiation. Yes. So my expectation, right, is that for the more elite, at least in this case, the expectation was we're going to have a much higher mean performance, but the genetic variance associated with that breeding with that breeding population was going to be lower. Obviously, that's based on Alice's experience working with these lines and the two different breeding pools. So that's where those expectations came from for the Mesoamerican and the Indian germplasm. I think that in general is going to be fairly crop specific. So will the sweet potato community model be used for other crops in Hawaii? Do you have suggestions for implementing this model for crops grown in New York State and beyond? That is a great question. So we have been busy drafting and redrafting and redrafting the cultivar release policy as it relates to native Hawaiian crops. The release policy for non-native crops is completely standard, modeled on the University of Florida and from the workshop that the PBCC did at NAPB like 5 or 6 years ago at NC State. What we eventually came up with, and I'm happy to share this with everyone, is at first it was very extensive, outlining lots of different potential interactions, and then it got cut. So basically now it is a 1-paragraph statement that says, due to this relationship between Native Hawaiians and Native Hawaiian crops. Each cultivar release associated with a Native Hawaiian crop is going to be vetted by basically a committee, and the committee is made up of Native Hawaiian scholars and community members. So from Department of Hawaiian Studies, we have a couple Native Hawaiian faculty members who are in ag who are on it, and then the dean of the college, and that's going to be the goal for doing that. A generalizable policy across the board was not something that we could find agreement on. And so the idea of having that be done in cooperation on a case-by-case basis seems to be the most appropriate way to do it, so that it becomes a true community back and forth with the university. So I don't know Cornell's history with, I guess it would be mostly the Iroquois Confederation in northern New York there, maybe Huron Confederacy, I don't know. But the university has a really bad rep in Hawaii. And so there's a lot that has to be restored in that interaction. So more talking is better. Oh, thank you, Rebecca. So others are asking about the interannual variations. So let's talk about the concept of yield. In the context of leaving yield on the table, should we be thinking of leaving value on the table? The sweet potato with low yield had high value to people, which could be taken seriously in the analysis. That is a great point. So we were seeing leaving yield and leaving value on the table in the context of these being commodities. And I think that you're exactly right. The value isn't necessarily, especially in the context of the sweet potato, the value didn't really come from the yield. And so, and in fact, that best yielder is just a source of carbohydrate, right? It's not really one that, it's really only, the only folks who are really interested in it are the brewers, because it's not great tasting, right? So I think you're right. The idea of what's going to bring value and what's going to keep a local breeding program going is a really good question. A big reason that that I'm taking the participatory approach here is because we don't have a lot of funds for stably doing things. And so the idea of helping a community work on a crop that they'll continue to breed, that they'll continue to grow these polycross blocks. I mean, you get people really into it, and you get basically the community on the station interacting with the university, interested in growing plants, and interested also in kind of maintaining that relationship. So for us, it's a way to work to release cultivars in a way that, even though any of those royalties might be minimal, the community impact is going to be bigger, we hope. It's, you know, I would love to take more credit, but I can't. It's Ted. So Ted grew up in Waimanalo. He grew up, you know, about 10 minutes away from the station. And this is a big thing that his program has been doing, that, you know, working in, he does a lot of work in sustainable ag. And, you know, for him, this is a huge part of the sustainability comes from the social component and making sure that everyone in the community still wants to engage with ag. And so I think that is correct. So yeah, I try to help. Hopefully that answered your question, Rebecca.
I think I should chime in here just to say we've already gone a bit over the time. It's fine with me, but I know, Mikey, I think you've got some time with grad students coming up for lunch. Okay. If there are no other questions, thank you so much. It was super fascinating.
Yeah, thank you guys. I really it's just a pleasure to be there, and I am happy to answer any questions, and so you guys just send me an email. I will hopefully answer right away.
This has been a production of Cornell University, on the web at cornell.edu.